Thank you all for joining us today. My name is Katie Maxfield, and today I'll be discussing DNASTAR software for Sanger sequence assembly and analysis. To start the webinar today, I'll be giving you some brief background on DNASTAR as a company, as well as our Lasergene software. And then for the majority of the webinar, we'll be doing a live software demonstration showing some key Sanger sequencing workflows. This will include an example of a de novo assembly, an example of a reference guided alignment, and there we'll also look at variant detection and analysis. And then finally, a third workflow called assembling groups. DNASTAR has been in business for over 30 years, and we've come a long way during that time. Founded in the lab of Dr. Fred Blattner at the University of Wisconsin, DNASTAR's commercial efforts were originally focused on DNA sequence analysis and molecular biology software. Now, as the fields of molecular biology, genomics, and biochemistry evolved, so have we. We now provide tools for next-gen sequence analysis, and most recently we've developed applications for protein analysis and structure prediction. This long history and wide use for applications has made DNASTAR the leader in citations in peer-reviewed journals. For the past 30 years, more researchers have cited DNASTAR software in scientific journals than any other commercially available sequence analysis software. In fact, earlier this year, we surpassed 50,000 total publications. If you want to learn more about researchers citing DNASTAR in their work, you can visit our blog and look at the publications category. Here you'll see a variety of usually monthly posts highlighting some recent publications citing DNASTAR software. So I encourage you to check that out if you're interested in learning more about some real-world examples of people using our software. And along those same lines, our blog also has a variety of customer interviews where we sit down with users of our software to learn how they're using our software, what they think of our software, and most importantly, learn about their research. So a couple of great resources on our blog. If you're interested, I encourage you to check that out by visiting our website and clicking on the blog link. DNASTAR is a global software company. We're headquartered in Madison, Wisconsin, but we have offices, distributors around the globe to supplement our in-house support team. And in our Madison, Wisconsin headquarters, we have a support staff as well as our development team. And we're able to then use any feedback you have to work with our developers to make our software even better. Our software supports a wide variety of workflows. This includes molecular biology tools for things like virtual cloning and primer design, multiple sequence alignments, and of course Sanger sequence assembly and analysis, which we'll be looking at today. For those users working with next-gen sequencing data, our Lasergene genomics suite integrates with our molecular biology suite and provides tools for next-gen sequence assembly and alignment, as well as variant analysis and gene expression. And then finally, our structural biology suite includes tools for protein analysis and protein structure prediction. All of this software is available on a variety of licensing and access configurations. We provide extreme flexibility in licensing, so you can use a desktop or network license, and all of the software is also available on the ground or on the cloud, which really allows you to take advantage of some unique configurations for storing and sharing your data, accessing your projects from anywhere, and as well as running some larger next-gen assemblies, for example, without investing in a lot of hardware. So that's just a very brief background on our company and on our software. And at this time, I'd like to jump out of the PowerPoint and move into a live software demonstration. Here I have the DNASTAR Navigator open on my desktop.
And the DNASTAR Navigator helps you to get started with DNASTAR software. And from here, you can access all of our applications and workflows. So today, we'll primarily be looking at the molecular biology tools, but you can also get to our next-gen sequencing tools, structural biology tools, as well as those cloud applications we talked about. So for molecular biology today, we're going to be looking at Sanger sequence assembly and analysis in SeqMan Pro. And if I hover over this, you'll see I get a little bit of information about the application, as well as links to some of those videos I showed you, direct links to our written tutorials, as well as demo data. So this is a really helpful resource for new users and especially users that are maybe evaluating our software for the first time. You don't even need to have data. You can use our demo data, or you can evaluate using your own data and use our tutorials and videos as a guideline. So we'll be looking at SeqMan Pro today. Quickly, I want to highlight Megaline Pro. This is an application for multiple sequence alignment and phylogenetic analysis. We will be getting to that later in today's presentation. And I'll also be showing you a little bit of SeqBuilder. So SeqBuilder is used for virtual cloning and primer design, but it's also a nice tool for sequence editing and annotation, as well as creating maps for publication. So to get started, like I said, we'll come to SeqMan Pro, so I'll just click that, and that will launch the application. So here I have SeqMan Pro open, and you'll notice I have two blank windows here. The first is the unassembled sequences window, so this is where we can add our sequence data. And then the project window, this will be populated after we perform an assembly. Before I do add any data, I just want to show you the project parameters. And so this is a variety of settings you can use both for trimming, assembling, contaminant removal, as well as some visualization, SNP options, things like primer walking. We'll be getting into all of some of these things later. And you can set these parameters before you get started, but most of them can also be changed on the fly. So for example, if you want to wait to do your SNP discovery parameters, you can do your assembly first and then set up some quality threshold parameters if needed. All of the parameters here for the various categories are optimized to be good for most general assemblies. For example, our assembling parameters have been extensively tested and optimized. So in general, we don't recommend changing these. If you do have an assembly problem, you can always call our support staff and we can walk you through some things to try. So I want to point out the parameters, but in general, you can just get started without having to worry about those. So let's start a project. The first project we'll be doing is a de novo assembly of some mumps virus sequencing data. And so here I have a folder of all the sequence traces. And so I'm just going to go ahead and select all of these and drop them into the unassembled sequences window. You could use the Add Sequences button too if you want to navigate to your files. So here you can see all of the trace files are loaded. At the top we see there's 300 total. And just to give you an idea of what the data look like, I'm just going to double click the first trace. And here you can see this opens up the chromatogram. So you can look through that and see the trace data. You'll notice at the end here we have some poor quality data. Always in SeqMan Pro you can trim things or edit things manually. But the better way to do this is to use the quality trimming that's built into SeqMan Pro. So if I were to just assemble right now, SeqMan Pro is automatically going to do quality trimming. But just to help you visualize, I'm going to walk through the trimming kind of step by step here. So what I'm going to do is select all of the sequences. And in addition to quality trimming, I want to perform vector trimming as well. So I'm going to set my vector to Janus. And you can set a reverse vector too if you want, but it's not required. And then what I'm going to do is click Options. And you'll notice we have some pre-assembly options, including trimming sequence ends and this is based on quality, as well as scanning for vector and trimming that away. So if you say scan later, SeqMan Pro will do this trimming as part of the assembly process. For demonstration, I'm going to click Scan All. And so now we can see the Janus vector for many of the sequences has a check mark next to the name. That means the vector was found and trimmed. For some sequences, you'll see an upside down question mark. That means the vector was not found. So let's take a look at this first trace again. Double click to open. And so then right away you see this grayed out sequence data, and that's the portion of the trace that was trimmed. And here you can see that's indicated as vector. So if, again, you can always come in and undo this trimming by manually dragging that sequence end. But what's nice is that that vector indicator stays there for you, so it's easy to restore that trimming. If we go to the end of the sequence, we see no trimming on this end. And also notice the sequence limits are updated, so you can get an idea from looking at these, how much data was trimmed away. And just a final look at trimming. The trim report shows the trim parameters that were used and how much was trimmed off of each sequence. So we can see, the sequence starts at 64. We can take a look at that. In this case, again, we have vector sequence trimmed and no trimming at the end. Let's say, for example, let's look at this sequence. No vector was found, but we still have maybe some poor quality data remaining. It's always a balance. You don't want to trim away too much of your data. But if you feel that maybe your data is a little bit messy, you want to trim more stringently, what you can do is go to Select All and Trim Ends, and let's set this quality stringency to high and Scan All. And so that's going to redo the scan, maybe trim a little more stringently, just to get any low quality trace data out of there. Generally, that's not needed, but I just want to show that you can do some iterative trimming if you need to, depending on your data. So now that we've looked at trimming, let's go ahead and assemble. And you'll see this report that gets generated here. That went pretty fast, but it just shows you all the sequences that are being entered into the assembly. And in this case, all of the sequences were assembled into a single Contig. And so we can just double-click that Contig to look at the alignment. And so here you can see each of the reads in the alignment. You can scroll along to see the results. Each read can also be expanded, again, to show the trace data. And like with the trimming, we have full editing capability here. So for example, this read, which is causing a gap, we can expand that to look in more detail. And if we feel that is, a true, high quality sequence that's causing that, we could look to, the other sequence data to see if there's low quality scores there. As the gap gets inserted, that's going to affect our trace data. So if you want, you can also look at the individual original data. And so you can see that that's nice quality trace data throughout. So this is just a way to help you sort of assess the assembly results, to look at any conflicts or gaps with some supporting data. One thing you may want to do with a de novo assembly is annotate the Contig. And we provide several options for doing that. And one is through BLAST. So I'm going to go ahead and get that started. So I've selected my Contig, and then I'll say BLAST selection. And this is going to perform a BLAST search on NCBI. And I'm just going to let that run in the background, and we'll look at annotation at the end of this example. So back to our alignment view, we can see our trace data, we can see the alignments, we can see conflicts in red. So this is a nice micro view of the alignment. If you want to look at the overall alignment, you can come back to the project window and say, right-click on your Contig and select strategy view. And so now we can see coverage along our alignment. And so we see kind of these peaks and valleys of high and low coverage. And this is fully integrated with the alignment view. So let's say we want to look at one of these high coverage areas in more detail. We can double click and jump right to it. Similarly, if we want to look at a low coverage area, that's integrated as well. So a few different ways to look at your alignment. In terms of the strategy view, you'll notice there's a couple elements here. First, this coverage threshold graph at the top of the window. And so this has either the thick green bar or a thin green bar or a red bar. And so the thin green means that this is below our coverage threshold, and the red bars mean it's above the coverage threshold. And so this is where those project parameters come in. So if you want to change how those high and low thresholds are defined, go back to project parameters, and under strategy viewing and coverage, I'm going to change the coverage threshold. I'm going to actually up that to 8. And just for demonstration, I'm going to change the maximum expected coverage to 16. And I'll say OK. And so now you can see this graph updates. So we have more of these red high coverage areas, and we also have more of the thin green bar area. I'm going to change the max back to 20 just to remove some of that red. So let's focus in on these low coverage areas. Again, you can double click to look at the alignment to see what could be going on there. But usually what you'll want to do if you're going to continue doing sequencing on this sample, you'll want to improve this coverage. So SeqMan Pro actually has an integrated primer walking tool. So what you can do is select your Contig and go to Contig Primer Walk. And so the primer walking allows you to design sequencing primers both to close gaps. That actually doesn't apply in this case because we have just a single Contig. But let's say the sequencing data went into multiple Contigs and you want to get sequencing data to fill in the gaps between Contigs and pull it all together. You could find primers that will help with that. In this case, we want to look for primers that will improve coverage. And we want to look for a minimum coverage of 8. So that's kind of matching our visualization parameters as well. So I'm going to leave the rest of the options at the defaults and click OK. And then SeqMan Pro generates a list of primers that are found along the Contig. And then if you notice, we'll zoom in. Those primers are also displayed on the strategy view surrounding those regions of low coverage. And again, all these views are integrated, so you can double click and then see the primer on the sequence in the alignment view as well. So that's some tools for assessing coverage and then designing primers to improve coverage. Let's come back to annotation. It looks like my blast search completed. So I'm going to expand this window just so you can see a little better. I don't know if you can read the text, but we have a lot of mumps virus hits. That's expected since this is mumps virus sequencing data. I'm going to sort this by query coverage so that all of the best matches are at the top. And you'll see we have a few different mumps strains at the top. So one option here, if you're interested in annotation of your Contig, is to pull out the annotations from these blast hits and apply them to your consensus sequence. So I'm going to select, say, the top 3 hits. You can do as many as you want, but just for ease of viewing, I'm going to select 3, and then I'm going to say collect features. And so what this does is takes the annotations from those blast hits and maps them on to my consensus sequence from the assembly. And so now we have this feature table that shows all of those annotations and where they lie on the consensus. So if we want to look at these in any more detail, we can just double click and jump right to that sequence. Oops. Let's try that again. There we go. Again, you can look at this in the strategy view. And so now you'll see these sequences are actually shown underneath the coverage graph, or rather the annotations are. And if you hover over, you can see more information about each of the annotations. You can also annotate manually. So I'm going to select my entire sequence. Just as an example, let's say I want to create a feature for the entire assembly. I'll just say features, new consensus feature. And I'll just say, name this mumps assembly. And I'll just call this a miscellaneous feature. You can add a note if you want. And so that's going to save that feature to the consensus as well. And so now let's say you want to save out this consensus sequence, perhaps for publication, make a map, or view the features, look at the sequence, etc. You can do that. You've kind of seen how you can look at it here in SeqMan Pro. If you want to do a little bit more, what you can do is select your Contig, or Contigs if you have multiple, and then say send consensus to SeqBuilder. And so this, I showed this in the Navigator briefly. This is our application for sequence editing and annotation. So here the default view is the entire consensus sequence. We can also display a linear map, and then we can add our features. So I'm going to add all of the features, and you'll see that adds all of the CDS and gene features, as well as some features SeqMan Pro created, and then the mumps assembly feature that I created. So that's obviously a lot of data. You can customize what features are shown. For example, let's say we want to look at our coverage features. So SeqMan Pro automatically defines these regions of above and below expected coverage, also areas that are covered on one strand. So you really have all of that data sort of concatenated down into one file. Let's just show genes and coverage below and the miscellaneous feature. And then from there, you can drill down further and say exactly which genes you're interested in. You can move features around, up or down on the map, and just really get this view customized. And then you can export this, say, for publication or presentation. So that concludes my de novo workflow. At this time, I can take any questions.
Okay, Katie, we do have a few questions that have come in from our attendees about de novo assembly in SeqMan Pro. And the first question is, does SeqMan Pro support mate pair data?
Yes, it does. That's a great question, and that's often very important for de novo assembly. I'll just briefly show you. So in this case, my project is using single-end data, and so in the bottom of the strategy view, you see these black arrows representing each of the sequences. If you have pair data, you can go to project parameters and set up your pair specifier naming pattern. And so then you can enter a sample of your names and verify that that pattern is recognized. And then what you'll see in the strategy view, you'll see green or blue or red arrows depending on if the pairs are facing in the right direction, if they're in the same contig or different contigs. And then you can start to use that pair data to do something called order contigs, and that will create a scaffold of all your contigs, basically using the pair data to start to pull things together into a single sequence.
Okay, thank you for that. And we have another question. Can other vectors, like somebody's own vectors, be added to SeqMan Pro?
Yeah, that's a great question. So when I showed the vector trimming, I just pulled a vector from the drop-down. That's using the default vector catalog. But you can, from the project menu, open vector catalog, and then you can add your vector sequence directly here. So you can just pull in your .seq or GenBank file of your vector sequence. And then once you've added it to the vector catalog, that'll be available in that drop-down menu so that you can use that vector for all of your SeqMan Pro projects.
Okay, that was all of the questions that we had for this section. So now Katie is going to move on to talking about reference-guided assembly.
Great, thank you, Sharon. So I'm going to go ahead and close out of my de novo project, and then I'll open up a new project, and we'll look at a reference-guided assembly. So in this case, I'm going to work with data from sequencing of a few different influenza virus strains, rather 2 strains. And so in this case, rather than working with trace data, I have FAST and qual data. So these are FASTA files with associated quality scores. And again, I'm just going to drop those in to my CMan Pro window. And since this is a reference-guided assembly, I'm going to also, at the same time, drop in my reference sequences. So this is the 8 segments of the influenza genome. So it's important to note that CMan Pro supports multiple reference sequences. You can also assemble against a single reference sequence for the entire genome. Also, if you're working on some kind of targeted sequencing project, you could drop in a reference sequence for the gene you're looking at, or you could drop in multiple reference sequences for each of the genes. Just depending on what you're working with, the software is flexible and can handle different setups. So I've dropped my sequences in. Now I just need to mark them as reference sequences. And then for my remaining data, we'll go ahead and do vector trimming again. So I'll select all, set the vector to Janice again. And now I'm just going to assemble and let CMan Pro do the trimming as part of the pre-assembly. And you'll see that assembly went pretty quickly. What CMan Pro does is creates a contig for each of the reference sequences. And again, we can look at our report just to see a summary of how all of the sequences were handled. We can look at the trim report to see quality and vector trimming data. And then just like in the last example, we can double click a contig to open the alignment. And so this time you'll notice, in addition to the sequencing data, we have the reference sequence here at the top. And so if we expand that, we can actually see the annotations in the reference sequence, if there are any. And just as before, we can scroll along the alignment to look for any differences, any areas of conflict or concern. For example, here we see some gaps. Now, this is FASTA data, so I'm going to display quality scores to look at the qual data that goes along with these FASTA files. And so I can see here, for example, this T, this end of this read has some, some base calls that are introducing gaps into the consensus. And this T, for example, is fairly low quality at 12. So I can either edit this sequence directly and delete that T, or I can just come in and delete the gap from the consensus, which is actually a little bit easier. So I'm going to undo, which is really handy. You can always undo any edits here. So I'm going to select that gap and just hit the delete key. And that also deletes the T from the individual read. And again, I have a T and a G here also causing a gap. None of the other reads show that, and those are low quality, so I could delete those. I could also trim this sequence, up to a portion where the quality scores are a little higher, and then remove any residual gaps. So, depending on what the data look like, you may or may not need to do any editing, but it's certainly useful to have the capability there. And like I showed you, you can always undo changes if you have any issues. So you'll see by default, the conflicts are shown in red. That's because CMan Pro is just treating them as differences from the consensus, or in this case, the reference sequence. If you're interested in variant analysis, what you can do is open up the SNP report. And so now CeqMan Pro kind of changes the view mode to highlight these differences. And so you'll see these blue columns of SNPs. And then this report, which is integrated with the alignment view, summarizes all the SNPs that are found. And so you can use the report to kind of toggle through. So you notice this 16%, this is just a gap here. But most of the SNPs are actually at 100% frequency. If you want, you can actually filter this to just show the 100% SNPs or 50% or whatever you deem the appropriate threshold. So to do that, you would click Filter, and I'm going to change the minimum SNP percent. We'll say 90 to allow for a little bit of variation. You can also limit by only substitutions or only indels. If you're looking for SNPs that are within a certain distance of a coding feature, you can use this filter, for example, to only show SNPs within 25 bases of a coding feature. You can also filter on depth of coverage, so if you're not interested in SNPs that occur in low coverage areas, you can easily filter that out. And then when you close this, you'll notice now I have 109 putative SNPs and 106 that CeqMan Pro filtered out. One other parameter you can adjust is the quality score. So just to ensure you are seeing more probable SNPs, you could filter only SNPs with a quality score of 15, for example. In this case, that doesn't reduce the total number of SNPs by much. We have 98 SNPs now. And so you could again look through the assembly at each of these SNPs. And once you feel confident that a SNP is a true SNP, you have the ability to annotate the reference sequence with a variation feature. And so you can do that on a per SNP or per variation basis by right-clicking on a SNP and in the SNP report, right-clicking and then saying Confirm SNP. Or you could select all of the SNPs and confirm them. And so that's going to annotate each of the SNPs, each of the variations along the reference as a variation feature. And so what that allows you to do is then save your reference sequence and use it for downstream analysis to compare SNPs between projects. Again, you can visualize these annotations in SeqBuilder. So if I look at my Contig and then send the consensus to SeqBuilder, you can build a new reference sequence here. Again, you can look at the coverage thresholds, and you can also see the original annotations from the reference sequence. But I wanted to highlight these variation features, so we'll just turn those on. So here you can see all of the variations along the reference sequence. And those will be saved with this reference sequence, as I mentioned, for any downstream applications you want to use.
Katie, is this a good time to forward you some questions?
Sure, yes, go ahead.
Okay, we have a few questions about the reference-guided assembly that you just showed us. The first question is, can I compare SNPs between samples?
Yes, you can. So let's come back to the SNP report for a moment. In this case, I have just I treated all my data as a single sample. So those are in the SNP report, just all together. Let's say you had a multi-sample assembly, which I'm actually going to show next. But in that case, you get one Contig per sample. In this case, we have one Contig per reference sequence. But you can compare SNPs between Contigs by opening up a SNP report for all Contigs. And so then that will show which Contig each SNP was found in. It doesn't quite apply for this example because this is treated as a single sample. But if you imagine the Contig ID being the sample ID, the SNP table provides one way to do that. The other option we have, particularly for people working with next-gen data, We have some additional tools for SNP analysis and filtering and annotation in our RayStar software. And so that allows you to set up a multiple sample assembly and then compare SNPs across up to very, large groups of, say, 100 or more samples and easily filter based on different criteria. So depending on, the type of data you're working with and how many samples you have, there's a couple different ways to approach that.
All right, thank you for answering that question. The next question is, can I change the consensus sequence to the majority call instead of the reference base?
Yeah, so in the alignment view, what Sharon's talking about is this consensus base in a reference-guided assembly always matches the reference sequence. Even if, in this example, every single read calls a C, but the consensus is an A. Now what I've done is annotated that A as a SNP. But what you may want to do is just override the consensus sequence. So what you can do is after your assembly, you can just unmark this reference sequence. And what that will do then is now use the majority call as the consensus call. So depending on how you want to use your consensus, you can go about that either way, either through annotation or through updating the consensus sequence.
Okay, great. And while you're here in the alignment view, we have another question regarding that, which is, why can't I see trace data for these samples?
Yeah, good question. So you'll notice before we had the little expandy arrows next to each of the reads. Those aren't visible here because what I imported was FASTA data. So in this case, I don't have the supporting trace data. What I do have is quality score information. So we're able to display that using this quality score tooltip, but the trace data in this case just aren't available. So whether you're working with trace data or FASTA data, you can import that into CMAN Pro, but the exact visualization of the read data will vary depending on that file type.
Okay, thank you for answering those questions, Katie. And now Katie's going to continue with the third and final part of the live demo, which is on assembly in groups.
Great, thanks, Sharon. So a question came up about comparing SNPs between samples. And that's actually a great segue into the next workflow, which is assemble in groups. So in this case, what I want to do is assemble, and rather than just assembling all the trace data together, assemble my samples into individual contigs for comparison. So I'm going to go ahead and pull in some cytochrome C oxidase sequences. And these are from different water mold species. And here I'm going to go back to working with trace data. So I'll drop those into the project. And so now, rather than just assembling these data all together, what I want to do is set up multiple samples. So you may have sequencing data, say from a 96 well plate, and you have 96 samples, and you want those each to go together to the exclusion of all your other sequencing data. So to do that, rather than click assemble, you click assemble in groups. And then you just need to set up your groups. And so this is all based on the naming convention of your sequences. So in this case, I have a rather long name, but I'm going to select the portion of the name that's unique for each group. And I could select this whole string here, or I can just select the last couple of digits. It really just depends on how you want to set this up. And as I make these different selections, you'll see the group name updating. So that's a good way to verify if you are, in fact, selecting a unique portion of the name. So I select 42, for example, and now my groups are 42, 43, 44, and so on. So once I've verified that my groups are set up correctly, I can click Assemble. And now I have multiple contigs, each with 2 sequences. And so now, rather than these contigs representing, say, a segment of the genome or a different gene, like in the last example, now each contig represents a unique sample. And so we can look at these samples, we can open the alignment view, we can open up the strategy view, just to, validate that the sequence goes in the reads go in both directions and you have good coverage along the contig. And we can do that for each of the sequences, and just to validate that we did get good sequence coverage in both directions. And from here, you could do SNP analysis. So I didn't pull in a reference sequence, but I could have. And then you could do SNP analysis and look at SNPs for all of the contigs. So in this case, we don't have any SNPs, but if you had a reference sequence here, we could start to compare SNPs between the samples. But what I want to do is build a phylogenetic tree of my samples. So what I'm going to do is I'm going to save out my consensus, and I'll just save this as a FASTA file. So I'm going to save this in my data directory. And now I'm going to go back to the navigator. And I'm going to open up Megaline Pro. And so this is DNASTAR's application for multiple sequence alignments. And this provides several different alignment algorithms that you can use. It also allows you to align both DNA or RNA as well as amino acid sequences. So what I'm going to do, I'm going to pull in that FASTA file that I saved. And I'll just specify this is DNA. And then we can go ahead and run an alignment. So I'm going to align, we'll use muscle. And so now these bars at the top represent each sequence, each consensus sequence in this case from SeqMan Pro. And the green regions are areas that match, and gray are regions that do not match. And you can look at that at this higher level as well as on a sequence level in this lower pane here. And so we can start to see very clearly some differences between the different samples that we assembled. You can also view a phylogenetic tree of these samples. You can see kind of two main clades here. If you want to build on this phylogenetic tree, say you want to add some other cytochrome C genes from different species to compare your samples, that's very easily done. I'm going to come back to my data folder, and here I have just a handful of sequences from GenBank. I'm going to drop those in. And so now these are added as unaligned sequences. You can view the annotations on those sequences here by expanding, very similar to what we saw in SeqMan Pro and SeqBuilder. And now I'm going to add these to my alignment. So I'm going to, first I'm going to select all the unaligned sequences and then say align, realign with selected unaligned. And I'm just going to leave all of the same default options and go ahead and run that alignment. And so now those gene sequences are added to the alignment. And we can see how they compare in the phylogenetic tree now. And this kind of supports the two clades that we saw before. So we have these 5 samples that group with these 3 sequences, and then we have our other sample that groups with these other GenBank sequences. And so you can build your phylogenetic tree that way. You can continue adding more sequences. If you had, say, more samples, you could bring those in from SeqMan Pro, or you could keep adding samples from GenBank just to build the tree of interest. You can also rerun the alignment using a different method. You can see it all happens pretty quickly, so it's very easy to tweak things and adjust just depending on your project and your data. So that concludes the assemble in groups. I can now take any additional questions that have come up.
Okay, we have a couple of questions, and they're both about data, adding data. The first question is, can I assemble next-gen data in SeqMan Pro? And what about a combination of Sanger and next-gen data?
Yeah, so you cannot assemble the data in CeqMan Pro. For that, we actually would point you to our CeqMan NGen application. And so that would allow you to bring in your next-gen sequencing data. And it's optimized to support a variety of different workflows, both de novo and templated and multi-sample that we showed today. But it also has some special parameters for the different next-gen data types. For example, Illumina and Ion Torrent have some different requirements in Sanger and from each other. And it supports whole genome assemblies, RNA-seq and de novo transcriptome, exome. So a variety of workflows, and it's just a little more fine-tuned to support those different workflows.
Okay.
And same with the Sanger data. If you're doing a combination of Sanger and NextGen, that would all stem from CeqMan NGen as well. But your analysis, I'm sorry to cut you off, Sharon. The analysis would be very similar to what I've shown today using the tools in CeqMan Pro.
All right, thanks for clarifying that. And the last question is, can I add sequence data to an existing project?
Yes, you can. So you can do that from CeqMan Pro using the sequence add command. And so you can bring in more data and assemble into your existing contigs or add new contigs. So let's say you want to only add new contigs, you can actually lock the contigs you've created. And then, like for a multi-sample assembly, you'd want to do that just to make sure you're not corrupting your existing samples. But let's say you wanted to actually add to these samples, you wouldn't need to lock them. In addition, you've seen here the assemblies all run fairly quickly, so you could just start your assembly from scratch and dump all your data in. So either way you want to go.
Okay, that's the end of the questions for this section. If anybody else has any additional questions, please go ahead and chat them in and we can answer them after the webinar. All right, so I wanted to show you a little bit about the DNASTAR website. We have quite a large collection of videos, and Katie showed you some of those at first, but I'll show you one that's related to today's talk. So you go to support and then videos, and you can see that they're currently organized by topic. I'm going to change that to product. I'm going to uncheck Array Star and check CeqMan Pro. And you can see that the second video down from the top is on Sanger sequence alignment and SNP discovery, which Katie talked about and did a demo of today. All of the videos that we have on our website, and there are probably a couple hundred, are just 2 to 3 minutes long. So they just give you a very brief introduction, but you can view them a couple of times if there's something you missed or you didn't quite see where, how we got to a certain screen. So those are really helpful to a lot of people. And now I'm going to show you our webinars page. If you're here at our webinar, you've probably already seen this page. Here's today's webinar. Here are past webinars that have been archived that you can watch. They're sorted by next-gen, molecular biology, and structural biology. And we're going to be posting some of our early 2016 webinars soon. So be sure to come back frequently and check this webinar page, and you can register for any of them by clicking on the register button there. So I also wanted to point out that we have a wide variety of written tutorials also for all of our products. If you work better with something written in front of you instead of a video, and again, these are organized by either topic or by product name. And you can click on those and get a nice written tutorial to follow. And we have the practice data available for download right here, so you can follow right along with those written tutorials. And let's see here. I have to unshare my desktop. There we go. So we just wanted to thank you for joining us today. This is the conclusion of our webinar, and we hope you found it very helpful. If you have any additional questions or you have some ideas for future webinars, you can email those to us at webinars@dnastar.com, shown here in the slide. And I just wanted to thank you again for joining us, and we hope to see you again for our next webinar. Thank you, Katie.
Thank you, Sharon. Thanks, everyone, for joining today.